Okay, so I'm going to be talking like to the camera as well, because we're recording and then putting on YouTube. So we're going to be reviewing for lab exam 4 for A&P1. This should be, I believe it's exercises 15 through 20 in your worksheets. So first, the slides are kind of all over the place. It's going to go like from anatomy of the axon to the brain, and then like at the end we'll get to the special senses. So it kind of follows the order that your worksheets go in, but it's a little bit all over the place. First, we're going to talk about cell types. So we have neurons. These are highly specialized cells that conduct messages in the form of nerve impulses from one part of your body to another. And then we also have the neuroglial cells. These are another major cell type in the neural tissue. These provide structural integrity to the nervous system and functional support that enables the neurons to perform. And then this is also part of what you need to know for your lab exam, the name of the cell and the general function. So the first one we have are the astrocytes. These are the most abundant in your central nervous system. So with these cells, you'll also need to know whether they're part of your CNS or PNS. Specifically, the astrocytes are found in your central nervous system. Again, these are the most abundant. These have a role in making exchanges between capillaries and neurons, synapse formation, and helping to determine the capillary permeability. These also control the chemical environment around the neuron by regulating ions and the concentration of neurotransmitters. Next, we have the ependymal cells. These line the CSF-filled cavities. These have a ciliated surface that helps circulate, again, that cerebrospinal fluid and form a somewhat permeable barrier between the cerebrospinal fluid and tissue fluid of the brain and spinal cord. Next, we have satellite cells and Schwann cells. These Schwann cells form the myelin and surround the neurons in your peripheral nervous system. The satellite cells, we can see it, let's see, up here. And then down here are the Schwann cells. The Schwann cells protect the neurons and aid conduction by forming an insulating cover called a myelin sheath, which is also something that you would have to be able to identify on the big Axon model. We have microglial cells. These are defensive cells in your central nervous system. They protect neurons by acting as macrophages to phagocytize any microorganisms or debris located around the neurons. So again, those are microglial cells found in the central nervous system. And then we also have oligodendrocytes. These have processes that form myelin sheaths around the central nervous system nerve fibers. So again, here are those myelin sheaths. And then they are covering the nerve fibers here. Okay, and then so specifically for those cells, you would need to be able to identify them by the image as well as their function. And then as well, being able to identify whether those are found in the CNS or PNS. Next, we have the Axon model that you're responsible for knowing. So this specific model will show up on this lab exam. We also have the stand-up model in our lab if you want to come look at it. So you will need to be able to identify the anatomy of this model. So first we have the cell body. These like little plankton looking things that are sticking out, these are the dendrites. And then down here is just the axon, which is myelinated. That's this blue covering here. Specifically, what's in yellow is kind of, you can expect it to be asked. Everything that's in white could possibly be asked, but everything that's in yellow is almost like for sure going to be asked. Inside of the cell body, we have the cytoplasm, which is similar to like a regular cell, an animal cell. Inside, there's these red little lines. These are the neurofibrils. And then we have our nucleus, and very inside our nucleolus. And then at the bottom, we have our nerve fiber bundle and our motor plate. getting a closer look at the bottom middle portion, we have again the axon, which is myelinated. Here's that myelin sheath, the covering. This large structure here is the Schwann cell. And then inside of it kind of looks like a raisin. That's the nucleus of the Schwann cell. This yellow covering inside is the neurolemma. And then this little divot here is the node of Ranvier, and then our nerve fiber. So again, we're just getting a closer look at the axon in this second image here. Again, anything that is on this slide is fair game for your practical and having to be able to label out. Okay, and then we have the tissue coverings of a nerve. This is very similar to like muscle tissue. This specific image is the one that shows up on your exam, and you need to be able to label it. So this very pointy, tippy top structure is just the axon. We have our 3 coverings. So the first covering right here, it's labeled number 2. This is our endoneurium. This second covering right here, number 3, is the perineurium. And then 4 is the epineurium. 5 is just covering the whole thing. So this is the nerve. It's just asking what the whole name of the structure is. 6, we can see it has a large circle around it, so it's talking about the whole structure. This is a fascicle. And then we're going back to the top where we have our axon label number 1. Number 7, we're going to identify number 7. That's the covering. This is the neurolemma. So again, this specific image shows up, and it was also in your worksheets. Next, we have the connective tissue coverings of a nerve. So number 1, these are our synaptic vessels. Number 2 is our axon terminal. Number 3 down here is our ion channel. And then this space labeled number 4 is the synaptic cleft. And 5 is our axon. Again, there's a similar image in your worksheet. This is one of the images you'll have to identify on your exam. Okay, and then we're moving into brain structures. So again, this specific image shows up on your exam. The way this is viewed is this portion right here is the top of your skull. So this right here is bone. I'm sorry, not bone, your scalp. This portion right here is bone. Here is your scalp. It has another layering right here. Right underneath that is bone, so your skull. We know that because of the compact bone in here. So right underneath our skull, we have our first layer of protection. That's our dura mater. So again, any impacts that's occurring on your skull, right underneath that is the dura mater. It does have 2 layers, but specifically we're just looking for dura mater. Right underneath that, we have our arachnoid mater. And then underneath that, we have our thinnest layer, that's our pia mater. So the way to remember the order of it, because this portion down here would be your brain, remember brain, pad. So it's going to be your brain, pia, arachnoid, dura mater, and then your skull. So brain, pad, skull. There's additionally another space that you're responsible for knowing. That's the subarachnoid space, which is in here. This is where the cerebrospinal fluid travels through, and it holds the cerebrospinal fluid in this subarachnoid space, which if we know our anatomy, we know this would be underneath the arachnoid mater. So again, this specific image shows up on the exam. Remember brain, pad, skull, and you know the order in which these sit in your brain. Okay, and then we begin to name the structures of the brain. So this isn't everything you need to know. I do have a couple slides of the brain, but it is pretty inclusive. First, we have our cerebral hemisphere. Let's see, our interventricular foramen in here. We have our hypothalamus here. Our optic chiasm is in here. And then right underneath that is our pituitary gland. Some of the more easily identifiable is our pons and medulla oblongata. So our pons right here, it's like a little extension. And then the medulla, which then the spinal cord. Going to the top, we have this big structure up here that forms two C's. That's our corpus callosum. Deep in here, we have our choroid plexus. And then we also have our thalamus down here that encloses our third ventricle, which we will talk about. We also have our anterior and posterior commissures. And then back here, we have our pineal gland, which is part of our epithalamus. And then moving into the midbrain, we have our corpora quadrigemina, which is right here, and then our cerebral aqueduct. So again, corpora quadrigemina, and then cerebral aqueduct in here. This entire structure here is the cerebellum, but specifically these structures inside of the cerebellum that kind of look like a tree. These are the arbor vitae. So on the exam specifically, you're going to want to read the question very carefully and make sure to identify what they're asking for, whether it's the entire structure, the cerebellum, or the inside landmarks or the inside structure of the cerebellum, which would be the arbor vitae. We have our fourth ventricle, and then our choroid plexus, and then again, our cerebellum. Okay, and then we get to talking about cerebrospinal fluid. You are going to want to know how to spell it out. It's at the top. And then additionally, on the exam, it may just be written as CSF. So you're going to want to also be able to identify that if they are, or if CSF is written on the exam, they're pertaining to the cerebrospinal fluid. So the composition is a watery solution. Compared to plasma, it has less protein and different ion concentrations. The cerebrospinal fluid does take on a constant volume, so it's always going to be the same. Functions of your CSF, it gives buoyancy to the central nervous system organs. It protects the CNS from blows and other trauma, and nourishes the brain and carries chemical signals. So in here, we have our ventricles. We have lateral ventricles. They're lateral because there's one on each of the hemispheres. So we have a left and a right. Our third ventricle in here, and then fourth ventricle. So a lot of students sometimes ask, why isn't there, or why is there a third and a fourth ventricle, but not a 1 and 2? The 1 and 2 are the lateral ventricles. They're just not specifically named 1 and 2. We just call them the lateral ventricles. So I do believe this image was also on your worksheets. You are going to want to be able to identify the different types of ventricles. And then again, this image was also on your worksheets. It's just showing you the ventricles again. You can see here the subarachnoid space, and then the different motors. And then the choroid plexus of the fourth ventricle. I don't know specifically if you need to know the path in which it travels, like this image shows, but you do need to be able to identify the ventricles from this image. And then some more labeling of the brain. Again, we have our cerebral hemisphere, our diencephalon in here, and then again, our cerebellum. Again, the arbor vitae being inside the cerebellum. And then what makes up our brainstem is the midbrain, our pons, and our medulla oblongata. Make sure on your exam, if they're asking specifically for this portion right here, that you're writing medulla oblongata and not brainstem. That's what I see a lot of students not get full credit for, is because they're writing brainstem instead of medulla oblongata. So again, medulla oblongata and brainstem are two different things, but the medulla oblongata is part of your brainstem. Some more anatomy. We have just another view. We have our frontal lobe, temporal lobe, parietal, and occipital. We do have our lateral sulcus, our cerebellum again, our pons in here, medulla, and spinal cord. On the lab exam, it's going to be really obvious what they're asking for. So they probably wouldn't give you a view like this to identify the pons, medulla, or spinal cord, just because it's a little bit harder. But any of the lobes, again, are fair game for the exam. Another thing or to keep a note for your exam is the view in which you're looking at the brain. So as you can see here, this model is like a cut view of a open skull, I guess you could say. You would have to identify the frontal part of the brain so that you can identify the right and left hemispheres. So we can see over here in this model, here is the nose. So we know that this is the front or anterior of the model that we're looking at. So our frontal lobe would be over here, making this side the left cerebral hemisphere and this side the right. So if we had it flipped and it was back here, it may change our view on things, but we always want to find the front of our model or anterior of our model and go from there, identifying specifically what we're looking for. So on this model, again, we have our left and our right, cutting it down the middle. That's why we have a corpus callosum on each side. We have our gyrus here. This long opening or slit is our longitudinal fissure. And then we also have our central sulcus labeled. This specific model, I do know that you looked at it in lab. I don't know what type of question they'll ask from this model, but these are identifiers that could come up. Okay, and then we start getting into our cranial nerves. So for your exam, you're going to need to know the cranial nerves by name and then also by Roman numeral. So the way I've seen it asked is they'll give you the Roman numeral and then ask you to write out the name of the nerve, or they'll give you the name of the nerve and then ask you to write out the Roman numeral. So I do think, yes. So here's 1 through 12. Specifically, you'll need to know, again, like I said, the name, but then you'll also need to know whether those are sensory, motor, or both. And so the mnemonic for that is some say marry money, but my brother says big brains matter more. And this goes in order. So some say would be sensory, olfactory and optic would be sensory. Marry money would be motor. And so oculomotor and trochlear would be motor. Center motor, yes. And then the first B goes with trigeminal, the trigeminal nerve. This is going to be responsible for both sensory and motor. So again, they can ask you name the cranial nerve indicated by Roman numeral 10. That would be the vagus nerve. And then, identify whether it's sensory, motor, or both. You would have your mnemonic here. Go find the number 10. That would be brains, vagus, both. And then there's a million mnemonics online for remembering the cranial nerves. The one that I like to use is on occasion, our trusty truck acts funny, very good vehicle anyhow. And that's just the cranial nerves in order. Again, it gets a little tricky with the 3 O's and the 2 T's right next to each other. the way that I remember the O's, you kind of just have to remember them in your head because those are very similar. Trusty truck, again, kind of difficult because they both start with TR. Trusty truck trigeminal is number 5. And the way that I remember it is some Geminis are born in May, and that's the 5th month. So that would be trigeminal number 5 truck. And then again, some say Mary, money, but then we would know trigeminal is responsible for both motor and sensory. And then here is the image in which you'll have to identify the cranial nerves. So we have first our olfactory nerve here. We have our optic nerve that has the chiasm and the tract. Oculomotor is right in here. See, trochlear right underneath. Trigeminal is our 5th, and that's right here. Then we start moving down. We have our abducens nerve. That would be 6. We're moving on to 7, which is our facial nerve up here. 8 is our vestibulocochlear nerve, which you'll also have to identify on the ear model. We have glossopharyngeal, which is 9. Vagus nerve is 10. Accessory is 11. And then hypoglossal is kind of special, kind of different, and it sits on top up here. So specifically, again, I've been asked the question, how do you remember abducens versus accessory and which comes when? Abducens is going to be 6, accessory is 11. AB comes before AC in the alphabet, so abducens is going to come before accessory. This specific image, again, will show up on the exam. So you're going to want to make sure you're comfortable with it, and then also be comfortable with all of the Roman numerals, spelling out each of the cranial nerves, and then also sensory motor or both. Okay, and then this is another way that the anatomy of the brain can be asked. On the exam, this is something similar to what you'll see. So A, up here, we have the cerebrum. B, if you can see it labeled right here, this is going to be our medulla. And then remaining from here would be the spinal cord. C is our pons. D is our mammillary body. Moving up, we have 4 up here. This is our thalamus. And then 5, we have the big C's. This is our corpus callosum. 6, this structure here is the fornix. 7 and 8, we can see they're kind of parallel to each other or across from each other. 7 is going to be the anterior commissure, and 8 is going to be the posterior commissure. 10 is going to be our hypothalamus. So again, we had our thalamus up here. 10 is going to be the hypothalamus. 11 is right in here. This is our aqueduct of the cerebrum. 12 is our tectum. 13 right here. This is going to be our 4th ventricle. And then again, 14, this particular number is referring to the arbor vitae. Specifically, this whole structure is the cerebellum. But again, 14 arbor vitae. And then if you can see it right in here, this number 17, this is going to be our oculomotor nerve. So again, this is kind of similar to what you would see on your lab exam. Okay, and then we start moving down in our body. So now we're going to talk about our nerves. So here we have our spinal cord and its nerve roots. We have up here our cervical spine, and then right here is our cervical enlargement. So again, cervical spinal nerves up here. Moving down, we have our thoracic spinal nerves. And then again, moving down our lumbar spinal nerves, and then our sacral spinal nerves. So it does follow the same, not pattern, but the same as our vertebrae. We have cervical, thoracic, lumbar, and then our sacrum. We also have cervical nerves, thoracic nerves, lumbar nerves, and then our sacral spinal nerves. Specifically, what you will be asked about is the filum terminale down here, our cauda equina, and then this conus medullaris, which I'm not sure if you can really see it, but it does have like this tapered kind of triangular shape to it. So that's your conus medullaris. Again, our cervical enlargement, lumbar enlargement, conus medullaris right here, the cauda equina, and then our filum terminale. Okay, I believe this image was also in your worksheets. This is the spinal cord and its meningeal coverings. So again, we have our spinal nerve here, our central canal in the middle. Right here, we have our ventral median fissure. And then we can see our spinal coverings here. So or the meningeal covering, sorry. We have our spinal dura mater, arachnoid, and then pia. So it follows that same order that we have in our brain. Pia, arachnoid, spinal being the most superficial. We have our white columns, which are just the white areas. We have our dorsofuniculus, ventrofuniculus, and then lateral. So again, knowing the orientation of the structure or model that you're looking at is super important. And then getting into more detail, our dorsal root ganglion here. Let's see, over here we have our dorsal horn, followed by our ventral horn down here, and then lateral horn, which is again lateral. All of these right here make up the gray matter, which is what you see right here. Like I said, we have our dorsal root ganglion. Over here, we have our ventral root. And that's what you need. Again, this is another model, something that you would see in your, or on your exam. Again, we have our gray matter here, our white matter, the posterior median sulcus, and our anterior median fissure, our dorsal root ganglion. We have our spinal nerves over here, our posterior funiculus, and then our posterior horn. And then down here is going to be the anterior horn. This just got in the way. Our central canal, and then lateral horn, and then our lateral funiculus. So again, this specific model does show up on your lab exam, and then you'll have to label the specific area that they're asking for. And then again, just another look at the different nerves. So this, what do we call him? This specific model also does show up. So we have our cervical plexus high up in our neck, our brachial plexus, and then moving down, we have our lumbar plexus here, and then our sacral plexus. And then we have our intercostal nerves, which are these right in here. And they're labeled. We have our musculocutaneous nerve, We also have the axillary up here. I'm not too sure if I have it labeled on here, but it is important. So we have our axillary, musculocutaneous, and then we get into our lower arm. So it follows the order in which, again, our bones are in our lower arm, forearm. So we have our radial nerve. When our model is in anatomical position, that's going to be lateral. Our median nerve, which is in the middle, and then our ulnar nerve is on our inside, closest to our body. And again, this is when we are in anatomical position. We can see on this side of the model that they are not in anatomical position because their thumb is medial or towards the inside of their body. So you're going to want to make sure you're paying attention to the model that you're looking at and make sure that they are in anatomical position. Moving down, again, these were our forearm, radial, median, and then ulnar on the inside. Moving down, here is our sciatic nerve. And then our femoral nerve up here. Okay, and then I do have it labeled over here. So this is going to be your axillary nerve, our radial nerve, and then again, median and ulnar. And then we also have our musculocutaneous nerve. These are just showing you kind of the divisions that you can see them. So here, the yellow is the anterior divisions, and then the green is going to be the posterior. So again, these are the major nerves of the upper limb and what you would need to know. Ulnar is toward or the closest to the inside of your body or the middle of your body. The median is going to be in the middle, and then the radial towards the outside or lateral. And then we get into the nerves of the lower body. So here they're showing you where the nerves are in relation to the lumbar vertebrae. So L1 through 5. The really important ones that you need to know are, again, the femoral nerve. The obturator nerve I highlighted because it is in here. It's pretty long, but it goes through the obturator foramen of the pelvis. So that one, if you can remember what this is called, you can remember the nerve. And then moving down, our saphenous nerve is also a long one, and that goes pretty far down our body. In here, we also have the iliohypogastric and then the ilioinguinal. But specifically, the obturator, femoral, and saphenous are important for our purposes. And then again, this is just telling you where it comes off of. And we can see the femoral nerve is coming off of L4 from the lumbar plexus. Looking posteriorly, we have our sciatic nerve back here, running pretty deep and long. We also have our superior and inferior gluteal. We have our common fibular nerve, which is going to be lateral because we know that our fibula is lateral. We have our sural nerve in here, which it is cut too, so you can see more of the nerves, but this right here is your sural nerve. Here is that common fibular nerve. We have our deep fibular nerve. Deep fibular is going to be right here, and then our superficial fibular. Lastly, we have our tibial nerve, which is just going to be posterior to our tibia bone. Now, make sure on your exam you're not just writing tibia, because technically that's the name of the bone and not the actual nerve. It's going to be tibial nerve. But if they are asking for the nerve, then just tibial would be fine. Okay, and then we have the basic components of all reflex arcs. So again, this is the big gray model, and it has the effector on it, and then it has like your the integration center on it, or the whole, it kind of looks like that spinal cord not structure, model. So first we have our receptor, which is going to be, in this case, our skin. And that's when there's a stimulus affected or something happened to the skin. In this case, the stimulus is stepping on a nail. We have our receptor, and then our sensory neuron here, which moves into our integration center, to our motor neuron, all the way to our effector, which would be our muscle. In here, we do have our interneuron. And specifically, I don't think I have an image of the reflex arc model on this PowerPoint, but we do have it in our lab, and it's that really big gray model, like I said. You do need to be familiar with that model as well. So I would also know the order that these occur. Okay, and then we start getting into the eye anatomy. This is something you'll need to know on both an image and the actual eye model. And I do have both of those labeled on here, so we'll go through those. This specific image, I believe, comes from your worksheet. So first we have our levator palpebrae superioris muscle. We have our palpebral conjunctiva, our cornea, and then the conjunctival sac. And then again, this is a lateral view of the eye that is coming from your worksheet. So this one you should have labeled already. We're going to identify the extrinsic muscles of the eye. I do believe on the actual lab exam, you'll do these on a model. Specifically the regular eye model. Let's see. So first we have our superior oblique muscle. And then right kind of underneath that is the superior rectus muscle. That one is directly sitting on top of your eyeball. On the side, we have our lateral rectus muscle. So this specific eye that we're looking at would be the right eye, because we know that this would have to be towards the outside. So we had our superior rectus up here, which means down here is going to be our inferior rectus. And then kind of cradling underneath our eyeball is the inferior oblique. Okay, now we have another view. This is going to be superior. So we're looking at the top of our eye. So again, we have the superior oblique muscle, the superior rectus here. Underneath it is that inferior rectus. And then underneath the superior oblique muscle, when we're looking at it in this specific, view, is going to be the medial rectus. And then the lateral rectus. So again, different views account for the different muscles. I would expect the different views that are on your worksheet to show up on the lab exam. Okay, and then we also have the lacrimal sac and the lacrimal glands. So here is the lacrimal gland. You'll also need to be able to identify this on the model. The lacrimal gland, we can see the pathway that it takes to the lacrimal sac, which then produces our tears. This is getting a little more into detail, but we do have the lacrimal punctum in here, and then the lacrimal canaliculus. I don't think specifically you'll have to identify these, but they are good to know. You will need to be able to identify the nasolacrimal duct, which is in here. And then our nostril, and then that's where all the fun stuff comes out of. But specifically the lacrimal sac and the lacrimal gland, you are responsible for knowing. And then as well, this image of the eye will also come up because it is kind of looking at something different or more structures that you wouldn't be able to see in the actual eye model. So I would be familiar with everything on the image, but specifically the things that are in yellow are really important and are things that are usually asked. So first, we have the ciliary body here. And then kind of directly underneath that, we have the suspensory ligament. This is our ciliary zonal. We have our cornea, our iris deep in here, and then our pupil. Specifically, we have the anterior segment, which contains the aqueous humor. And then back here is the posterior segment, which has the vitreous humor. So you will need to know which of these contains which type of the humor. So anterior has the aqueous and the posterior has the vitreous. We also have our lens here. Moving on, we have our sclera. This is also known as the whites of the eyes, and it may be referred to the whites of the eyes on your actual exam. We have our choroid right here. We can see it is both right here directly underneath that sclera and then also pulled back here. And then we have our retina, which is inferior to the choroid. And then deep in here, we have our macula lutea here, which is this circular portion. And then if you can see, there's a little bit of a darker spot in here. That's the fovea centralis. So on the exam, I'm not sure which of these they'll ask about, but you do want to make sure you're identifying whether they're pointing to the specific entire structure or just this little pit inside, which would be the fovea centralis. We have our optic nerve back here, which is one of our cranial nerves. And then deep in here, we have our optic disc, which is also known as our blind spot. So again, you do want to be familiar with this entire image, but everything in yellow is what we see asked a lot on the exam. Okay, and then here's, again, another view of the eye anatomy. So this is kind of what you'll see, or the type of model you'll see on the exam. Yeah. So back here, we have the optic nerve. We have our superior oblique muscle, the medial rectus, our inferior oblique muscle, which again is kind of hugging the actual eyeball or the sclera. And then this colored portion here is the iris. Again, here's another view of the eye anatomy. Here's that lacrimal gland I was talking about that you're responsible for knowing. We have up here our superior rectus muscle, our lateral rectus. Deep inside here is our medial rectus, and then underneath is that inferior rectus. And then back here is another view of that optic nerve. Again, a superior view. We just went through these, but just so you're familiar with the different types of views. So medial rectus deep in here. From this specific view, we can see it more in this one. Since they do use this specific model, I would be familiar that the superior oblique is kind of up here hiding. We're not able to see it on this one, but it is up here kind of hiding on this specific model, which we also have back here in the lab if you do want to take a look at it. I do know some of the classes did the, what is it called? The dissections. Not every class did, but it is possible that may show up on the exam, as well as the sheep brain. So you just do need to be familiar with what you would be looking at in a dissected eyeball. So here we have our cornea, this white portion is supposed to be the sclera. We can see it's not as white as it would be in a human or what it looks like in our images, but that's what that would be. Here again, the sclera. We have the retina deep in here, the pupil, cornea, and then the entire lens. Specifically, if they're going to ask you something from one of the dissected specimens, it's going to be really obvious what they're asking for, just because it is a little bit harder to identify some of the structures on a dissected model. Okay, and then we get into our ear anatomy. So we have our outside of our ear here, our auricle, or it's also referred to as the pinna, the helix, and then the lobule, which is your earlobe, or kind of where you get like that first ear piercing. This long kind of opening or canal is that external acoustic meatus. And so the external acoustic meatus is still part of your external ear, which is this whole portion here. This whole portion is your external ear. Our external ear and our middle ear are separated by a specific structure. This is the tympanic membrane. This also comes up as a question, like what structure separates the middle ear from the outer ear? The tympanic membrane. Within our middle ear, we have our auditory ossicles, which we'll talk about. And then we have our innermost portion of our ear, or internal ear. And then this houses the pharyngotympanic or auditory tube. And then also our cochlea, our semicircular canals, and the nerves. So again, we do have those three auditory ossicles. We have our malleus, our incus, and our stapes. We have, again, that pharyngotympanic tube here. And then specifically, you will need to be able to identify the auditory ossicles. So I know the last time we talked about them, you just needed to know the names of the ossicles. Specifically now, you do need to be able to identify them. The stapes is usually the easiest to identify, just because it looks like, I'm not sure the correct term, but like the horse thing that you step on. The malleus and incus, I always just remember the malleus comes before the incus, and it's kind of directly on that tympanic membrane. So again, for the auditory ossicles, if you remember MIS, that's the order in which they can be viewed from the ear. And then we start getting into, again, that internal ear. So we have our semicircular canals. There is an anterior, posterior, and lateral portion of the semicircular canals, but you're just responsible for knowing semicircular canals. We can see our stapes down here. And then this structure here is our cochlea. Oh, not that. So we will get into more of this anatomy, but we do have our vestibulocochlear nerve coming off of here, and as well our facial nerve. So this is those structures of the ear I was talking about. Here we have our cochlea, our vestibule here, the oval window, and that stapes that's articulating right here, and then our semicircular canals. So as long as you can remember that these circular structures are called the semicircular canals, you're good to go for the practical. We also, so this specific nerve here is going to be the vestibulocochlear nerve. There is 2 divisions to this specific nerve, but in all, you just need to know vestibulocochlear nerve. Again, here's another look at that tympanic membrane. Again, that separates the middle ear from the external ear, and then attach the malleus, the incus, and then the stapes. And then here's how that sits within our ear. So this is a superior view. Here's that external ear, the pharyngotympanic tube, our tympanic membrane. malleus, incus, stapes. Here's that vestibule, our semicircular canals, the cochlea, and then that vestibulocochlear nerve back here. Specifically regarding the cochlea, it does open up in our models to show you the organ of Corti, which is that really big green model. I don't believe I have a picture of it on the PowerPoint, but it is in our lab, and as well, it will be on your lab exam, and it's showing you how the sounds that we hear or that we produce kind of flow through our cochlea. So the inside of this right here is pertaining to that big organ of Corti model. Okay, and then this is regarding the cranial nerve 1, olfactory. So we have that olfactory bulb, and then the tract, and then how that pertains to smell. And then this is one of the last things that is covered on your lab exam. I believe this is in your very last worksheet. It's just knowing the anatomy of the tongue. So knowing the specific anatomy of the tongue, and then how the cells, like the gustatory cells, where they're at within our body. So I would be familiar with the, what are they called? The gustatory, these are the gustatory cortex in your worksheets. So on our tongue specifically, we have the foliate papillae here on the sides. Right here, our fungiform papillae. This is looking at the taste buds, and then this is a larger view of that. And then this specific image, again, we have our pons, our solitary nucleus in our medulla. So this is showing you how the specific nerves take our taste to our brain. So specifically, I do believe you'll need to be able to identify these 3 nerves. So this one on the outside is going to be the facial nerve, or number 7, glossopharyngeal, which is going to be 9, and then vagus, which is 10. So these are the 3 specific nerves that you will need to be able to identify. And that's it. We do have the practice exams available, and they'll be available until the last exam is taken, which I believe will be on Friday.